So we also want to consider RNA in our discussion because it's a type of nucleic acid. And to start to look at RNA, we can actually compare it to DNA because there are some similarities, but there are a lot of differences, and we want to look at what those are. So the first difference, as you can see in this chart, is that the sugar is different in RNA. So it's a ribose. So on the second and third carbon atoms, we actually have hydroxyl groups, and that was different than in DNA. In DNA, we saw a hydroxyl group on the third carbon. There's also differences in the bases, so instead of having thymine present in this molecule, we're going to have uracil. RNA is single-stranded. However, it can base pair within itself and with other RNA molecules. However, it doesn't form a helix and it is not self-replicating. A lot of the RNA that we have, we understand it to be functioning in the cytoplasm. There is some RNA that's found in the nucleus as well. There's going to be a lot of different functions that are given to RNA. Classically, we've thought that it mediates transmission of the genetic code from DNA to the cytoplasm where it's made into proteins. However, we're finding that there's many different types of RNA molecules, and those different types have different functions. The length of the molecules are different as well. When you think of DNA, there's coding regions, there's non-coding regions, there's regulatory regions, and pieces of DNA that are in chromosomes are ginormous structures. However, RNA is very, very short sequences in comparison. As well, RNA is a very unstable molecule compared to DNA. Part of it is because the ribose sugar has hydroxyl groups bound to the second and third carbons in the ring. Also, we have RNases. So RNases are enzymes that actually degrade RNA. And so when you work with RNA in a laboratory setting, you have to make sure to try to sterilize and kill any RNases. We actually have lots of RNase enzymes on our skin because a lot of viruses, their genetic material is RNA, and so it's a protective mechanism to be able to degrade the RNA that would be in viruses that might be detrimental. So here, if we start to look at a DNA versus an RNA strand, you can see some of the similarities, some of the differences. So similarities, we have cytosine, adenine, guanine, but here's a difference. Instead of thymine, now we have uracil. If we look at the sugars, we had a deoxyribose, now we have a ribose. That ribose means on the second carbon here, we have another hydroxyl group. We have the same type of linkages, though, that form the sugar phosphate backbone. So we have this phosphodiester linkage between the 5' carbon of one nucleotide and this third carbon on the next nucleotide. So single-stranded RNA, if we look at its primary structure here, we have the nucleotide sequence. While it doesn't form helixes, these primary structures, if there's complementary base pairs, will actually form structures. They will turn and they will base pair on themselves. And so areas where we have these straight, nice areas, this is a tRNA molecule. Here's another straight area where there's base pairing here. That's called stems, because it looks like a stem. And then where you do not have base pairing, so here in this big circle, this is called a loop because there are no base pairs that are formed. And what we're finding is that, just like we say in many other areas in biology, different structures give us different functions. So hidden in this primary structure of the RNA molecule is a code for how to form a secondary structure. And with each different secondary structure, we can have a different function for the molecule. This ability to have different types of RNA actually lends complexity to eukaryotic cells that we were not aware of when we were not detecting these different types of RNA molecules. So traditionally, what we have thought is that there are three types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. And we've seen that in eukaryotic and prokaryotic cells. However, in eukaryotic cells, now we're seeing that there are many different types of RNA molecules if you start to look at this list, and they each have different functions. And so I'm sure in your careers you're going to keep seeing this list grow as we start to detect the different types of RNAs and explore their functions. This chart gives you an idea of some of the functions of these types of RNAs that you might not have seen before. So up at the top we have the traditional types of RNA that most people are familiar with. So ribosomal RNA is actually a component of the ribosome that helps to convert messenger RNA to proteins. Messenger RNA, remember, that's the carrier. And so it's the link between the nucleus and the cytoplasm. It's literally the template that's made off of the DNA molecule. And then we have transfer RNA. So transfer RNA is going to facilitate bringing in amino acids into that ribosome and matching them to the codons that are present in the messenger RNA. Now these new types of RNA, you can see that they're all small, and that's probably why we were not aware of them before, because these are tiny, tiny fragments of RNA, and some hide out in the nucleus, some are out in the cytoplasm. And so thus far we know that they do help in processing. They can help with the processing of messenger RNA, they can help with the processing of ribosomal RNA. Some, like the small cytoplasmic RNA, functions seem to be variable. It acts in different ways. And we've actually harnessed these last two, microRNA and small interfering RNA, which we call siRNA. If you've ever heard of a knockdown, when we knock something down in a cell, we actually put in small interfering RNA. And what these two molecules do, the microRNA and small interfering RNA, is they're a short fragment of RNA that actually can bind to the messenger RNA. And when it does, it forms a double-stranded RNA molecule, and that's a signal for enzymes in the cell to degrade that RNA. And so as it does that, then this is giving us a layer of control, a layer of complexity in the cell. As you decrease the messenger RNA that's encoding for a particular gene to make a particular protein, now you're regulating the production of that protein. So you decrease the amount of that protein if you use siRNA. And that's why we call it a knockdown, because you're knocking down that protein. You're not going to have as much functional protein.